It feels like there's not enough room in my body for my ovaries right now. Like I can feel how big they are every time I sit up or laugh or sneeze. It's a really weird feeling.
After 10 days of injecting myself with hormones, my ovaries had swollen to the size of 2 big, juicy lemons. This was to be expected. The drugs had revved up my reproductive system so that doctors could retrieve as many eggs as possible from it. The days of hormones had made me bloated and exhausted and cranky. But it was the trigger shots that really got to me. After giving myself a final 2 shots that would make the eggs I was growing all release at once, I felt almost like I had the flu. The big day had arrived. The day of my retrieval. Or, as I called it, the harvest. I had fasted since midnight and arranged for my friend to pick me up after the procedure. Stu was still living in Scotland, but had frozen his sperm a few weeks earlier, after we'd agreed to move forward. Now I was just ticking through the to-do list I'd been given one last time.
Let's read through this sheet of paper. So it says, do not eat after midnight. Anesthesiologist will review my medical and surgical history and begin an intravenous line for sedation. The egg retrieval procedure takes approximately 15 minutes from start to finish. So all this has been leading up to 15 minutes.
I was feeling nervous. Nervous because I'd never had any kind of surgery before, and at this point in the process, it was clear that freezing eggs or embryos is a serious medical procedure. I was also nervous because I didn't know if it would work.
I know I'm not going to get like 20 eggs because I don't even seem to have 20 follicles, but I'm hoping that there's like 10 good mature ones. I think that's like a pretty conservative hope. I think at the max, maybe I could get like 15 or 16. At my age, that would be amazing. That's like, you go ovaries, you go.
This was one of those cases in which knowing a lot about the topic did not make me feel any better.
Will my eggs be good quality? And then even if they're good quality, will they mesh well with Stu's sperm? Because there's a possibility that I get 16 eggs and we don't even get 1 embryo. That seems unlikely, but it's possible. Or, if we only get 1, then I'm going to have to go through this whole thing again.
I'd arrived at NYU's big gleaming fertility center on 53rd Street. The half-hour cab ride had not settled my nerves.
So, I'm just getting changed to go in. I can't bring my phone or my recorder in there, so I think this will be the last dispatch until after.
I was about to find out whether this ridiculous gamble was going to pay off.
Feeling nervous. Really very nervous. Okay.
If you're an extremely type A control freak, like me, the next part of the whole process is the hardest. I'd done everything I could to stack the odds in my favor. I'd researched clinics and picked one with a good reputation. I'd improved my diet and cut out booze and caffeine. I even took prenatal vitamins. I mean, it couldn't hurt. Now, the fate of my frozen potential future children would be in the hands of lab techs. The part of the process patients don't get to see. But also, maybe the most important part. I'm Kristen V. Brown, and this is Misconception.
Yeah, so this is embryo number 1. See how pretty is this? So I'm gonna go in and suck it up with my pipette.
I'm in the lab of the Manhattan branch of CCRM, a national fertility clinic chain. Emanuela Molinari is walking me through her day-to-day. She's an embryologist and the lab director.
So you put the embryos into those little wells? So
Yes,
they're
ma'am.
both in the first well?
Nope, one of each, because I need to know which one is what. I'm doing genetic testing to them.
What you're hearing is Emanuela very carefully sucking a couple's embryos out of a petri dish in order to biopsy them and send them for genetic testing. I cannot emphasize enough how tiny these things are. Barely visible to the naked eye, if you have really good eyesight, which I do not.
Now I'm using a big magnification to see very well all the structures of the embryo. See, this is the ICM, the compacted cells that are going to make the baby, and these are the trophectoderm cells. These are the cells that are going to make the placenta.
And you could not see anything with your naked eye on that?
Nope, it's as big as a grain, so we need to magnify it 40 times. to be able to see it this way.
Emanuela had moved the embryos onto this crazy-looking microscope that was outfitted with a laser and these two really fine needles that she could control. I was watching what she was doing on a video screen. A grainy gray mass of cells, all bubbly and lumpy.
So now I'm just gonna blast my laser and I'm going to zip it. See?
This has to be one of the craziest things I've seen in my reporting career. Emanuela uses this tiny needle to poke a hole in the side of the embryo and then suck out just a few cells.
I use this technique, it's called flicking technique. I don't pull it, I'm not a puller.
And then, with a laser, she just lops those cells off to send them in for testing.
You understand it? It's a little bit like playing video games, right?
Yeah, totally. So that's the laser.
Yeah, and boom, done.
Wow.
This whole idea of assisted reproduction, it can feel so high-tech. But what I was realizing was that it's extremely manual. It's Emanuela and her colleagues carrying around embryos in a cramped lab with music pumping. They're doing a lot of things by hand, checking each other's work as they move stuff around in a lab to make sure there are no mix-ups. This is a side of the whole process most patients don't get to see. They talk with their doctors, but they don't see the lab. They probably don't even consider the role that people like Emanuela play in their quest for parenthood. Emanuela walked me through some other parts of her day, like freezing embryos.
Concentration of water decreases and the cryoprotectant gets into the embryo. It will start to slowly sink down to the bottom. And look how it looks now, all crunched up. It looks like a cookie.
At the end of the day, I was struck by how much skill and finesse her job requires. Emanuela isn't just a scientist. She's an artist. I talked to William Schoolcraft, the founder and chief medical officer of CCRM, about the role the lab plays in a patient's success.
A friend of mine made a list of all the variables in the IVF lab. In one IVF cycle, there's 200 different variables. There's 35 ingredients just in the culture media. Then the media, which is the liquid the embryo is grown in, is overlaid with oil. There's protein in the media. There's the petri dish. There's the instruments called the pipettes that handle the embryos. There's the quality of the air, the settings of the incubator, on the list goes. And any of those factors, let's say a petri dish that had toxicity to an embryo, if you put good media, good protein, good oil in, and you put an embryo in, but the dish is bad, the embryo will get killed.
The finesse of an embryologist isn't even the only variable in the lab that matters. Another fertility doctor I spoke with said his clinic discovered the oil on the door hinges was impacting their success rates. And so now they use plastic hinges. Embryos are so fragile. Keeping them alive outside of a human womb, it's hard.
It's a game of perfection. And I would say it's just being very careful to not induce stress to the embryo. So the less we can stress the embryo, the more we can make the embryo think it's in the human fallopian tube growing in nature, even though it's outside the body, in a petri dish, in an artificial incubator, with different pH and CO2 and osmolarity, the better we're going to do.
I was feeling stressed about my embryos being stressed. It was starting to feel like it's a miracle that this ever works at all. We'll be right back. As I was waiting to hear how all my microscopic eggs had fared after my retrieval, I started thinking about what had brought me here in the first place. The time on my biological clock was running out, and I was in no position to start a family. It's not an uncommon story these days. It's a big part of why both egg freezing and IVF are booming. But neither of those things can really stop your biological clock. Existing reproductive technologies can press pause on your fertility or give it a little boost. But once your eggs are too old or gone, that's it. When I looked at success rate data on IVF over the past 20 years, I was shocked to see that rates had barely improved. For women over 42, the number hadn't really changed at all. A doctor who had been involved in the data analysis explained why to me. She said that if you're infertile, there's just only so much technology can do for you. There's a limit to how successful IVF and egg freezing can be. I wondered then if anyone was trying to address the root cause here, the rapid aging of human ovaries. That's when I met Daisy Robinton.
Our ovaries undergo rapid decline, after age 30, 35. And so I became obsessed with this idea of how our ovaries age at an accelerated pace.
Daisy is a Harvard PhD, and she teamed up with two other scientists from Massachusetts General Hospital David Pepin and Pat Donahoe. They started a company that is actually trying to slow the aging of the ovaries. Their company is called Oviva. The key to that aging, they think, might be a hormone called AMH.
AMH controls our ovarian reserve and how many eggs, are leaving at any given point in time.
You might remember AMH from episode 1. It's a hormone I got tested to try and figure out if I was still fertile. The idea here is that if you can regulate AMH, you could control how fast or slow those eggs leave the ovaries. You could slow that aging down. Eventually, they want to create a drug that you could use to slow ovarian aging to extend your years of fertility or even delay menopause indefinitely. Their first drug target is for patients not responding well to fertility treatments.
If you take a garden hose and you put a kink in it, it blocks the flow of water. When you release the kink, there's a spurt followed by a normal flow of water. And the AMH drug works sort of like this. You get this spurt, so this wave of eggs that are released all at the same time. That's greater than normal. You have a greater number of eggs you can actually capture.
Generally speaking, more eggs mean better odds in the fertility world. A lot of doctors and scientists I spoke to for this series mentioned to me that it's been more than a decade since there's been any radical shift in the science behind IVF. Daisy's co-founder, David Pepin, was one of them.
Frankly, it's kind of shameful. There are a number of women's health indications, like, say, polycystic ovarian syndrome or endometriosis, that are huge, affecting a large proportion of the population that have been understudied for so long. And so it's part of a general trend of women's health sort of being ignored. And hopefully we're starting to see some signs that this is changing and there's more attention being paid.
It actually seems like we're at a really exciting moment. This growing interest in fertility is just now starting to lead to new science and technology that could really move the needle. Oviva is a young company. So far, David has just shown the effect of AMH drugs on cats and mice. They're still a few years away from clinical trials in humans, let alone having a drug on the market. But things are happening.
I'm actually quite hopeful. There's actually quite a bit of research that has been going on, sort of slowly in animal models that could translate into massive, leaps in terms of reproductive technology if they were applied to humans.
Researchers today are investigating things like how to grow immature eggs in a petri dish, which could increase the odds of IVF. And scientists in Japan are even trying to make eggs and sperm from stem cells. That would be a game changer, not just for patients dealing with infertility, but also LGBTQ families. So far, they've been successful in mice. They made pups from 2 mouse dads. Robotics and artificial intelligence could bring improvements to IVF clinics and labs even sooner. A company called Overture is one among several that is working to automate some of what an embryologist does, which could help eliminate human error. Many more are using AI to try and fine-tune processes, including dosing medications and selecting the best embryo. So much of human reproduction is still a black box, and we are really just starting to peer inside of it.
All right, are you there?
Yep, I'm here. Can you hear me?
Okay. Yep.
The day after my egg retrieval, my doctor gave me a call to see how I was doing.
All right. How are you feeling today?
Just low energy, but otherwise actually better than before the procedure.
Good.
Yeah.
Yeah. Are you taking any pain medication?
I've been taking just 2 ibuprofen every 5 or 6
Honestly,
hours.
even though I was still extremely bloated and tired and, constipated, I was elated. I'd gotten 16 eggs. That was more than I had ever imagined. I was pretty proud of my now 37-year-old ovaries. But also, I knew that this was just the first stage in a long battle of attrition for my little eggs. Now, I was going to find out how they fared at the next stage, fertilization.
Are you ready for your results?
Yes.
You did great.
Of my 16 eggs, 14 were ripe. 1 was underdeveloped, and another, as my doctor put it, was overcooked. 13 of those fertilized. So, in 24 hours, I had already gone from 16 eggs to 13 embryos. And only 11 of those were completely normal.
This is an awesome start, or I guess continuation. Yesterday was a good start. It's a good step 2.
We were going to have to wait a bit longer to find out where we really stood.
So I will have all of the information next Saturday for you in terms of what made it to the embryo stage. And then the genetic testing takes another, 7 to 12 days after that to come back.
Okay, cool. Well, thank you so much.
You're welcome.
Seems very positive so far.
It's good. I'm super happy with the results so far. Have a great day. Bye.
Okay, bye.
That 2 weeks was agony. I am not a patient person. It also gave me a lot of time to think about everything that had transpired. So far, it was going well. But I was feeling kind of burnt out and kind of broke. Even with my great insurance, I had been hit by more than one unexpected medical bill. I still couldn't help but feel like somehow winding up in this position was my fault. I felt guilty, like I had taken a wrong turn somewhere. I read a book by Yale anthropologist called Motherhood on Ice by Marcia Inhorn. In it, she interviews 150 women who froze their eggs. And when I read it, it was striking how many of them had stories like mine. It was cathartic. She concluded that there's a mating gap, that society had changed in a way that just made it harder to start a family earlier in life. 82% of the women in her study were single, with no partner in sight. The rest were either in unstable relationships with men or waiting for their partner to be ready for parenthood. In a way, egg freezing is a technological solution to what is ultimately a social problem. When I talked to her, she said she thought egg freezing is sort of a stopgap, something that works for a lot of people, but is an imperfect solution. I feel hopeful, though. Our understanding of female biology is changing every day. In the future, maybe single 30-something women won't have to go through any of this. Or maybe, if they do, at least their insurance will cover it. After 2 weeks of waiting, my doctor called with our results, our final count of embryos. Stu was in Edinburgh, so I called him to share the news.
So, are you ready for it?
Yeah, what's the news? Tell me.
It's good news.
That's good.
We got 4.
Four like that are healthy and have all the right stuff in the right places.
Yep. So we don't have to do it again.
Oh, I'm so pleased for you, especially because I know it's been really bloody tough.
We had 4 embryos, 3 boys and 1 girl.
So how do you feel about it?
You know, it's really good to know that we have that sort of option there. These little frozen embryos are now sitting there and, you know, you could pull them out and use them at whatever point we decide. Well, I'm glad you told me.
I was thinking about not ever telling you, actually. I was thinking about just like withholding this information. Yeah, just making you wonder.
I mean, I would have asked you at some point, like, you know, whatever happened? Have you heard back about that?
Since we froze embryos in July, a lot has happened. Stu moved to New York and we got married. Kids still feel a bit down the line, but I think in the end we're glad to have our little embryos on ice. Our journey still isn't over. We might use those embryos one day. That is, if we can afford to keep paying for them. The day after I finished this script, I got an email from our fertility clinic. They were raising our embryos' rent. This series was written and reported by me, Kristen V. Brown. It was produced by Gilda DeCarli and Stacey Wong and edited by Cynthia Coons. Additional research was done by Tanaz McJohnny. It was engineered by Blake Maples. Our theme music was composed and performed by Hannes Brown. Special thanks to Shelley Banjo, Randy Shapiro, Anna Mazarakis, Jeff Grocott, Laura Zelenko, and Creighton Harrison. Sage Bauman is Bloomberg's head of podcasts. Thanks for listening.